Sandoz, thank you very much for that kind introduction, and thank you all for coming to listen, and the Society for offering me the opportunity to share with you a program which I think is quite exciting and brings opportunity to people for whom an unmet diagnostic need and the potential to stratify healthcare may be the result. So I'm going to talk to you about the 100,000 Genomes Project, and I'm going to focus somewhat on cardiovascular disease. I forgot to put in a disclosure slide, but I'm seconded most of my time to a company owned entirely by the United Kingdom taxpayer called Genomics England. So the origin of this project is unusual because its origin is not in a grant application as anybody wrote. It is in fact, this man on the left-hand side here convened a summit at the London Olympics, and he gathered together some important people, and these important people came by unorthodox routes and entertained us greatly, and eventually it was suggested to him by an august body of international scientists that the time was right for the United Kingdom, with a free at the point of care healthcare system, to undertake a massive program that would drive the application of genomic medicine into clinical practice here in England. And so this is him announcing the project in December 2012, and I will now describe to you about the project. But it owes its origin not to a grant that any of us wrote. I didn't write any grants for this in terms of its formation. Its origin is here at the London 2012 Olympics. So we're sequencing 100,000 whole genomes in patients from the NHS. So what I want to try and explain is that this is not a research program in its own sense, other than that there is research in it, but this is actually an NHS transformation program. So what we're doing is we're saying to the National Health Service at a point when we're still exploring the area, that if we position the research cheek by jowl with the healthcare system and we get this right together, then we can simultaneously transform the application of this in the NHS. And this is focused on disorders that are tractable to this technology. So this is rare inherited diseases where there's a clear inherited basis, cancers where we can use this to stratify healthcare, and also infectious agents where knowledge of the genetic architecture of the organism may help us to predict antimicrobial resistance, response, or even design a new group of drugs that we could use to treat these infections. It's whole genome sequencing, it's not parts of the genome. So some of you will have heard of exome sequencing. This focuses on the 1% of your and my genome that actually codes for proteins. And this is focused on reading as much as we can of the 3.3 billion letters that make up your and my genetic code. These make you the individual you are, along with your lifestyle. They also bring you some of your talents, which you inherit in some measure, and also allow you to have susceptibility to disease. We're used to thinking of populations, but I'm going to describe to you and focus in on rare disease today because that's of more interest to this society. And that actually means our usual focus as a group of people is very different, because then these people become live as individuals and families who are affected by these diseases. So our ambition of the program is to generate health and wealth, leave a legacy of infrastructure behind, and the human capacity and capability to deliver this program at scale in the National Health Service, and to be nothing short of world leaders in the application of genomic medicine in the healthcare setting, all by the end of 2017. So no pressure at all there. So I'm going to focus on rare disease now, and I won't speak anymore much about cancer or infection because I felt the Society would be more interested in this area because of our past history of interest in rare syndromic forms of blood pressure. So although rare diseases are individually rare and may only affect 5 people in 10,000, that's the European definition, because there are 7,000 or more, they are actually quite common. In fact, there's 3 million people in Britain today with a rare inherited disease. So that's about 5% of the UK population. We're currently working now, we've enlarged this from 128 to about 140 different disorders at present time, which have been nominated by the National Health Service, by academics, to work on. And these are undergoing very detailed phenotyping that's initially clinically primed. And the reason for this is because when I came to this project, I realized that for some of the people actually not getting a diagnosis, the characterization of the patients, because there was no diagnosis that could be made, sometimes was somewhat superficial. And so if we're going to take on the challenge of the most difficult cases, we need deeper phenotyping. So we've created deep phenotyping models for all the diseases that we work on, including all their diagnostics, their prior genomic testing, imaging, pathology where appropriate. And because we need to share this data with people around the world, because there might be only one family affected by a rare disease here in the United Kingdom, we've anchored this in a commonly used human phenotyping ontology, which is used worldwide to classify these diseases. That means that my classification of a disorder here in the United Kingdom will be the same as others around the world, so we can share data because we'll have data commons. We're also developing metrics of disease progression, because I found in some disorders that they simply classified the progression as mild, moderate, and severe. And I think we can do better than that. And if we're going to have genotypes and follow these people up longitudinally, then and see what genotype influences their outcomes, then we must, by definition, have a better categorized system than that. We're reading the genome at 30x. So what I mean by that is, imagine that you're reading a book and you come to a paragraph which is a bit challenging, difficult to understand. What will you do? You will read and reread till you find the meaning buried in that paragraph of what the author is trying to suggest to you. So this is what whole genome sequencing is like. Because we want to see rare variation, we need to read the genomes many times. So we read the genomes up to 30 times, and I'll show you our data on how well we're doing with that, so that we can see rare things that are hidden in the genetic code. We're also building a national rare disease registry. I mentioned a moment ago the importance of subphenotypes. If you see along the bottom line of this graph here that there are 10 different subphenotypes that characterize the disease, and you look at the different lines, if we only have 1 in 10 or if we have 100%, if you compare those two, my ability in terms of power is dramatically increased the more of those subphenotypes we collect, such that 10 allow me to have almost 100% power, if I have enough families with that disease, to be able to detect a rare variant. This doesn't always work, but this does suggest to you that just collecting 2 or 3 of these will not be enough to get us to a diagnosis. So how are we doing? On the left-hand side, you see a description of how well the sequencing depth is that we're achieving currently. So we're reading across the genome about 34.5x, so slightly better than 30x at the moment. And on the right-hand side, what I've done is to do what you would want to do, which is to ask the question, how good is this compared to a test I can do in the healthcare system? Because if we're going to adopt this, and it will be expensive to do so, we have to be able to justify the health economics. So on the right-hand side, you can see the coverage of all 600 genes in the UK genetic testing network that are currently available for testing in clinical genetic service labs. And all but 2 of the 600 genes are extremely well covered, read at greater than 15x depth across the genome, and 95% of the genome is read. So this is impressive. And the 2 that are not read are because they have highly repetitive regions. So I'm going to move now to share with you some of the first diagnoses that we've returned to patients. And this was done in March of this year at the Centre for Life in Newcastle. And this is the second, well, you won't need me to tell you who that bloke is. This is Patrick Chinnery here, and Paul Brennan, who ran the centre. And this is the Secretary of State meeting a family who's just received a diagnosis. So I'll take you through some of these families, and I'm going to focus on one who had a cardiovascular presentation but has a renal disorder. So this is a family with inherited focal segmental glomerulosclerosis. And the father, who's this case here, presented in his 20s with very high blood pressure and already quite advanced renal damage, such that he was on dialysis age 29, had a renal transplant age 30. His transplant failed when he was 55, and he was retransplanted last year. And his father and brother shown here, and his uncle not shown in this family tree, had all died of the same condition. Unfortunately, he's transmitted it to his daughter. His daughter now has high blood pressure and protein loss, but because her blood pressure has been extremely well controlled, she has not progressed to renal failure. So she has normal renal function right now, except for protein loss in the urine. Now, she's been worried sick for the 15 years of her daughter's life that she has transmitted it to her. And what she's been doing is getting her daughter to go along every month and have blood pressure and urine checks at her GP. So we found a known pathogenic mutation in a gene called IMF2, inverted Forman 2, that had not been detected because it had only recently been discovered and had not been analysed by the local lab. So this allowed this gentleman who had the kidney transplants to get his diagnosis. We found it co-segregated in the family. He transmitted it to this lady. But the most important thing that we've been able to do is that the NHS, having confirmed this test, then went on to test this lady in the middle. And this has been the source of worry to this mother here. And this lady doesn't have this mutation. So she no longer has to have blood pressure checks and protein checks. She will never get this disease. So this is one of the most important things you can do in medicine, which is to provide reassurance to this lady here. Here's a second group, not a cardiovascular group, but a family of two brothers who presented with increasing difficulty walking and turn out to have a distal motor neuropathy. Their symptoms began late in life, so rare diseases don't have to present in childhood, though the overwhelming majority of them do. And what happened is these became progressive. If they came in here and walked across the front here, they have markedly abnormal gait, and they would have difficulty even getting across the room. They're 79 and 72, so it hasn't shortened their life. Their mother and their maternal aunts all had this condition. We discovered a new mutation in a known gene for this disorder, causing a premature stop codon in the serine palmitoyl transferase transporter gene. And this means that they cannot transport serine properly, an amino acid in your and my diet. This was Sanger confirmed in Newcastle by the National Health Service, and they chose to decide, once they'd proven it co-segregated in the family, to feed this back to the brothers. Now they have a potential to participate in a pre-planned trial of L-serine supplementation for others with this condition, because we now know their cause. Now, let me describe to you the cost of this. This isn't health economics, but not getting these men a diagnosis costs the National Health Service £2,905 in serial testing, which essentially did not reveal a genomic diagnosis, with nerve conduction studies that simply said, yes, I'm afraid your nerves are a bit worse. For far less than £2,905, in fact, in each case, close to £700, we were able to bring them a genomic diagnosis. So this potentially, it's not health economics, but it potentially indicates that in some cases we can offer something that might save money. Now, we've just in the process of opening our new Barts Heart Centre, which is a 60,000 metre squared building on the site of St Bartholomew's Hospital, and we've moved 3 hospitals, London Chest, UCLH Heart Hospital and the Barts Hospital into this building. And a big focus here will be on inherited heart disease. So I'm now going to share with you the phenotypes that we're working on. So a number of these will be well familiar to you. So there's familial forms of aneurysm here. There's various disorders of rhythm, cardiomyopathies. And yes, as you would expect, I've made sure that extreme early onset hypertension in childhood or in the early life is included as a disorder, because we do find people coming through clinics and paediatric service with unexplained hypertension at a very early age. We will also be looking at some of the structural heart diseases shown here. These are more challenging to do and will require larger numbers. So I'm going to describe to you where we've got to with implementing the program very briefly. So what we did first of all is we had to come up with a way of involving the patients in the NHS and the clinicians. So I came up with a concept of genomic medicine centres, and NHS England has commissioned 11 of those and is looking at commissioning another few at the moment. And these are centres of excellence that will enrol patients according to a broad consent, supply us with the clinical characteristics and the appropriate samples. They flow into the NIHR Biosample Centre, which is south of here in Milton Keynes, and then once they pass quality control, into a sequencing centre which has just been completed at the Sanger Centre. It's a Sanger building, but one floor is the NHS first sequencing centre, and that was funded by the Wellcome Trust. With our partners Illumina, who produce the best quality product in the world, and we surveyed all the competitors by the way, generate whole genomes, which is combined with the clinical data in an identifiable database. This identifiable database can be enriched from primary care data, hospital episodes, outcomes, and all the registries that we have, including a patient entry portal, which I'll come to in a moment. This will enrich that data set, and this data set faces individual clinicians can access their data in identifiable format for clinical care. So this project faces 2 ways. It faces the National Health Service returning diagnoses for validation and transmission to patients, and then we pseudonymise the data and push it into a £24 million data centre which we've built, and I'll show you where that is in a moment. What this allows us to do is to present the research community and those in training and industry with sequential bills of pseudonymized data associated with the whole genome sequence. And it creates a safe haven within which now 32 is 35 clinical interpretation partnership domains, which I'll explain in a moment, can work together on the data and drive up clinical interpretation because we link it back to the identifiable data. So new diagnoses will come from the research engine that will be transferred back to the health system. And this is where all of you are here in this section here, coming in to use the data and to generate new findings. We've also contracted with a number of annotation suppliers, companies that provide services. This is extremely complex to do, and there's no gold standard for analysing the data, so we have to get help. We've also formed a partnership with the FAR Institute and the Oxford Big Data Institute to drive up the quality of this real-world data so that we can use this increasingly in healthcare studies to do investigations, not just in genomic medicine. So we've awarded 11 GMCs, as I mentioned a moment ago. This is our biosample centre here, that's the Minister for Life Sciences, and this is our sequencing centre here at Hingston. And this unimpressive grey building is in Corsham in Wiltshire, and it sits on top of a 4,000-person nuclear bunker, which is the government's nuclear bunker, and it's where the government keeps most sensitive information. It's inside a military base, and that's where our data is stored. Actually, one really neat thing about this data centre is because it's on top of a quarry, they bored multiple holes through underneath. So what happens is when the racks generate heat to store the data, they suck cold air up and self-cool through the tunnel system, so we don't have to spend money on air conditioning. And here are the genomic medicine centres, regionally distributed, with only 2 regions missing at the moment, Yorkshire and Avon, from the group. We've also funded, in addition to that, 530 person years worth of master's training at the cost of £25 million across 9 HEIs to build the capacity in the framework. Now, this is ResearchKit, which is an Apple app which was launched on the 14th of April. By the middle of May 2015, having been launched just 1 month earlier, 35,000 people had used this version of the app to enroll in a study at Stanford University. Now, you and I could never achieve that, but we can with this technology. And this is an example of the same type of smartphone technology. Simple things that adjust your screen, can tell your position and posture, and measure tremor. So if this is Parkinson's disease, measuring 4 Hertz tremor in a patient with Parkinson's and recording their activity level, because the phone can tell when you move. So this is the type of thing that we need to do more remote monitoring. And we're building patient-reported outcome apps for the diseases we're working on. So the patients can also tell us more information about how their things are going. And so we can share findings with the patients in general terms. So we formed a clinical interpretation partnership, and these are the funders that have committed money to it already. Now, normally when you and I do medical research, we do what we've been hearing about for the last 24 hours or so, which is to form an idea, get a group of people together, get some funding, start to collect data, analyse it, validate it, and then publish it. And then we attempt to get the health service to adopt this. This can take, in some instances, up to 17 years. We can't do that with this. We have to drive it into care much more rapidly. And if we get this right, this will become the paradigm for doing more fundable research at scale of the type you and I want to do. And so what we've done is to assemble a coalition of the NHS, academics and trainees right at the outset, which goes well beyond a research collaborative. Over 2,500 senior clinicians and scientists have volunteered to be in this partnership, and it's all UK-led. And they will then validate, publish their results, and then become champions for this, to drive it into healthcare, associated with health economic validation, so evaluation, so we can have the evidence to inform NICE and others, and all within 3 years. So these domains are multidisciplinary teams of researchers with the NHS. So if you submit your families that you've identified for many, many years and followed and looked after, you can be involved at the heart of the analysis as an NHS clinician. But you can also be there with many other skill sets that will allow you to drive up clinical interpretation. We've also embedding a stratified healthcare and therapeutic innovation domain, which will look at all the proteins coming out of the program in rare disease and cancer for druggability, look to see if there are small molecules that could be used to drug these targets, and then identify novel targets or repurposing opportunities from things sat on the shelves in pharmaceutical companies. We're also now in advanced planning to embed clinical trials in embassies where private clinical trials will be run on the condition that the data at the outset, the baseline sequence, goes into our data centre with the clinical characteristics, and at the end of the trial, all the data goes into the data centre when the trial is reported. This will allow us to attract people and new medicines in cancer and hopefully in infection and possibly in rare disease to the UK so that patients get the benefits first. So this is really hard to do. It's very, very challenging. If you imagine sifting through one genome is 3.3 billion letters. So we're aware of the challenges. We're working with NHS, academics and industry to make this a success. We'll support it with education. And at the end, the aim is to leave a legacy that allows the health service to do this routinely and to bring new diagnostics and therapies to the most important people in this program, which is not you and not me. It is, in fact, the patients. And also, just to say briefly, I'm extremely close, within probably days or weeks, of bringing Scotland and Northern Ireland into the programme. So it's Genomics England, focused on the English NHS at the moment, but Scotland and Northern Ireland will be coming in shortly. And these are the people involved who I should thank. This is the board of the company. You'll recognise some of the names there, and we've various advisory committees. And this is my team, so you can tell we're not spending the money on me. Thank you very, very much for this opportunity.
Thanks, Mark. Any questions? So, Mark, I have a question. I'd like to ask your opinion on the current thinking about privacy and confidentiality. In this era of big data, when genomics is one, when you start integrating a couple of data sources, people say that you can say goodbye to privacy because you can actually identify individuals through different methods.
Yes, so we are very upfront about this, Sandosh, and I think we should all be to patients. We tell the patients in our informed consent literature, we will do everything we can to protect your identity, but the depth of this information means that it would be potentially possible for somebody to identify you, and that possibility remains. If you talk to the patients who are enrolling the patient in the pilots, and 50% of the people with rare disease enrolled, and 90% of patients with cancer, they don't care about that. What they want is from you and I is a diagnosis. Indeed, when we presented the results to the family with the focal segmental glomerulosclerosis, the media tackled the family about the privacy and were they worried that we would share their data with people around the world. And the man faced the lady from Sky News down and he said, look, I've got a diagnosis for the first time. My family have got a diagnosis. My granddaughter doesn't have this. I wouldn't have got this if I didn't share my data. And so I have this disease and I want to share my data. So, it's up to me. And that is a general attitude. However, that is not the attitude of all people, and it's not the attitude of certain data lobbyists. And so we do have to be extremely careful. And the thing that keeps me most awake at night is maintaining public trust and confidence, which, fingers crossed, so far we've managed. But that's always fragile and should never be taken for granted.
Mark, I assume you're not concentrating solely on what you regard as single gene disorders, if that's still an okay term. If you're prepared to look at disorders where there's more than one locus contributing, how easy is it going to be to recognize which the pathogenic locus is? Because if there's more than one locus, then there'll be lots of unaffected people with it. And often the difficulty is knowing whether your patient does have a single gene disorder or not.
Yeah, you're absolutely right. And even within a disease, you can have compound heterozygotes. So you can have two mutations within the same gene that individually would not give you the disorder, but the combination of the two have a deleterious effect, which cause you to get the disease. So identifying those can be also challenging. So some of the disorders are rare diseases which would be more towards the oligogenic spectrum. So if you think about it, Maurice, if you look back a couple of years, it was 6,000 rare diseases. So as we get into this, ostensibly hypertensive phenotypes that we think have similarities will turn out, like Gordon's disease, to have several different loci, and we have to be open to that possibility. How do you solve this and make sure that you resolve it down to a causative mutation? Well, again, this comes back to what Neil says earlier. In fact, medicine is largely probabilistic. So you get to a certain point in causation where you've got the best possible potential of a disease-causing variant, and that's the point at which you call a diagnosis. So how do you filter these things down? First of all, you go to a certain number of trios, and if you have two parents and one offspring, you can filter out a lot of rare variants because of shared inherited bits bits of the genome which are not present in unaffected individuals. If you have a project at scale where 50,000 genomes are spent on rare disease and 33,000 of those genomes are on parents, you can use the scale of the population to filter out rare variants. So you use a sequential, if you like, filtration process to focus on a few variants, and then eventually you would have to do some degree of functional characterization in addition to standard bioinformatics. So you are right, in some diseases we'll end up with a handful and be unable without additional functional characterization to get further. That's why we're also taking on every single participant multi-omic samples. So the NHS is drawing RNA PAX gene tubes on germline, and they will be doing it on cancer. They're drawing serum and plasma for proteomics, metabolomics, and so we will be able to combine other omics to try and sift down to the rare variants of interest.
Mark, fascinating. You're working closely with industry, which is excellent, but of course industry has its own motives.
Yes.
Let me briefly give you a story which troubles me, and I don't know how it relates to what you're setting up. Fabry's disease is an unusual cause of stroke. The company will do the genetics of it for free because the intervention is horrendously expensive.
Yes.
Now, there is good evidence or reasonable evidence for renal and cardiac protection, but the data that patients who are younger patients who've had a stroke get any benefit from recombinant enzyme is pretty minimal. And yet the company are manipulating us. And I just wonder how you deal with that kind of issue in your system.
So at the moment, all the companies that are working with us, the 12, are in a pre-competitive consortium where they can't take away anything they own. Genomics England owns the intellectual property. We'll readily license it to third parties. But also we're looking at models in our IP agreement for benefits to the NHS. So discovered here means cheaper drugs for the National Health Service, research to be done here in the United Kingdom to evaluate that drug so that we can know that our scientists and our patients have the best chance of making the first observations and also bringing it into care. So we are linking the industrial involvement to benefit for the NHS directly. And in fact, that's a real focus of George Freeman, the Life Sciences Minister. How do we accelerate medicines in, but also use the work that we've done on those medicines to extract greater benefit for the health care system, for the reasons you say?
I suppose the subsidiary question is how do we make industry do the trials that follow on? Just because something's missing or overactive and you do something, you replace something that's missing, doesn't necessarily mean that it will have any effect. We do need to test that.
Well, of course, you're absolutely right, Philip. Industry obviously have the agenda of producing the medicine. They start with the patient benefit at the heart of it, but they're also interested in profit. So they have a focused series of deliverables. I think that's where you and I come in. And in hypertension, where industry in terms of drugs has largely left the building and we're left with device-based therapies and industry there, then I think we have to do more of those in the affordable way Tom showed us earlier. And the practical reality is that requires a coalition of all intellects in this society to make the unanswered gaps be answered. And that will require us to use public and charitable funds to plug the gaps that industry won't.
So, thank you, Mark.
Thanks very much.
For opening this exciting new era in genomics.